All right, good morning, everyone. This is the FES Center, University Hospitals, APT Center, Neuroengineering Center, all these different things, Neuroprosthesis Seminar. We're very, very thankful to have special guests this week.
Hey, Andy. How are you?
Mike Moffit is here to join us, and he is now a bigwig at Boston Scientific. So Mike and I have a long history together, and I think we were part of one of the more interesting and exciting times in the history of kind of neuroengineering at Case. And so the late 90s, early 2000s, there were an influx of all kinds of interesting and diverse graduate students that have now gone out and had very successful careers in academia and industry, and so we had Marone Bixen not long ago visit us, Mike and I, Raphael, who's now some kind of a VP at Medtron or at Boston Scientific. And yes, sorry, that was bad. And so all of us were students together at the same time, and it was kind of a funny experience, actually, I think, because we were all from very different backgrounds. So Mike and I were Warren Grill's first graduate students, and well, lots of people in this room probably don't know, I was a strange character in my younger years that Dr. Mortimer could tell you about. And so here was this, alternative skateboard punk, sharing an office with Mike, this very staunch conservative Mormon, and somehow we became like really good friends. So that's kind of, the nuances of graduate school. So anyway, Mike did his undergrad at BYU, and then obviously his PhD, his master's and his PhD at Case and BME, and then Mike and I worked together again later on at the Cleveland Clinic, and then Mike went on to work at Advanced Bionics, which then was acquired by Boston Scientific, and Mike continuously rose the ranks there. And now his title is Director of Neuromodulation Research and Advanced Concepts, and he's going to tell us a little bit about doing neuroengineering in a big multinational corporation. So thanks.
Well, I am getting closer to needing a wig, but I don't know that I'm that big. But I appreciate the kind words, and I appreciate the invitation to come and speak with the group, because it's been a fun couple of days and a fun trip down memory lane for me. I lived in Cleveland from 1998 till about 2005, if I include the postdoc. And I've been at Boston Scientific from 2005 to present. So I'm going to talk today about Boston Scientific's SCS and DBS systems. Those are the two franchises we're in. I'll spend a few slides on Boston Scientific and just orient the group to what Boston Scientific is like organizationally. But I'll get pretty quickly to the technical capabilities, because that's what I like to talk about. And I'll spend most of my slides on that. And then finally, Cameron asked me to spend a few minutes talking about what it takes to go from concept to product, what's involved. So I'll spend a few minutes on that as well. Okay, so who is Boston Scientific? Well, Boston Scientific's a large organization. They have divisions or business units all across the country and facilities worldwide, as Cameron mentioned. The number of employees, I don't know the exact number, but it's probably somewhere around 25,000 employees worldwide. And each of these business units supports different medical technologies. So these core businesses are shown on the slide here and include interventional cardiology, cardiac rhythm management. And that team was purchased from Guidant in the, I think 2007 timeframe. It's up in Minnesota. Endoscopy, neuromodulation, which I'm going to spend the bulk of my time speaking about. Electrophysiology, urology, women's health, and peripheral interventions. So all sorts of medical devices. So Boston Scientific Neuromodulation is a team that was originally part of Advanced Bionics, which was a startup funded by Al Mann. And Advanced Bionics, if you're not familiar with it, it grew up as a cochlear implant technology. And sometime in the late 1990s, I believe, I wasn't there then, but in the late 1990s, they were looking at expanding their capabilities and putting their technology in other applications. And spinal cord stimulation was one of those applications that they chose to pursue. In 2004, Boston Scientific acquired Advanced Bionics. And there's a long and interesting story about the acquisition and how everything sorted out. But in the end, the pain management business, which is spinal cord stimulation, and the deep brain stimulation business stayed with Boston Scientific. The cochlear implant business went its way, and Al ended up selling it to a different company. The team is located, the office is in Valencia, California, which is 2 valleys north of LA. It's about 30 miles north of LA, so that's a 2 to 4 hour drive, depending on traffic. The 2 business units or franchises within Boston Scientific Neuromodulation are spinal cord stimulation, which is done to treat chronic pain, and will be the example that I use for most of my talk today. Boston Scientific or Advanced Bionics entered SCS in 2004 and launched their SCS2 system in 2005. And then deep brain stimulation was something that was always of interest to the team. And finally, in 2012, we entered the market in Europe only. So we have EU approval only to treat Parkinson's disease and dystonia with our deep brain stimulation system. So I wanted to quickly just orient folks to spinal cord stimulation. I remember when I was a student, we talked about a lot of different stimulation applications, but spinal cord stimulation wasn't one we spent a lot of time on. So here I'm showing a transverse section of the spinal column. And so you can see the spinal cord, and the leads are actually placed epidurally. So this is the dura here that I'm pointing to with my mouse pointer. And then the cerebral spinal fluid lies between the neural parenchyma and the dura. So you can be anywhere. Your leads are actually anywhere from a millimeter or so to several millimeters from the neural target. You can also see we've pointed out here the dorsal roots with the red arrow. The dorsal roots are important because they're typically associated with side effects. So when you stimulate in the spinal column like this and excite the neurons of the dorsal columns, then you generate a sensation called a paraesthesia. And the paraesthesia is manifest on different parts of the patient's, the pain patient's body, depending on where the electric field is located. If the dorsal roots are excited, what you'll see is stimulation of the abdomen typically, just because of the rostrocaudal position of the leads. And usually patients don't like that. So in spinal cord stimulation, the goal of the therapy traditionally is to place paraesthesia over the pain topography and to avoid stimulating the dorsal roots, which would give you abdominal or uncomfortable stimulation. So in a nutshell, that's spinal cord stimulation. Of course, it started in the late 1960s with the work by Mortimer and Reswick. And I believe back then, and I'm not the best person in the room to tell the story, the electrodes were subdural. But as the therapies advanced, it's gone to an epidural placement. And these leads that I'm showing you in cross-section are isodimetric and are placed through a TUI needle into the space. There's another type of lead that I'll show you in a minute called a paddle lead that requires a neurosurgeon to place it. And something on the order of 50% of the leads are surgically placed, and about 50% are placed through the TUI needle. All right, so I want to orient you briefly to the precision spinal cord stimulation system components, just so that you're familiar with them as we walk through the talk. I mentioned the leads. The leads look something like this. These leads on the bottom are isodimetric. They have, the ones that we use have 8 to 16 contacts, so I'm not showing the 16 contact lead here, but have 8 to 16 contacts. And the one on the top is our surgical paddle lead called Artisan that has 2 columns of 8 contacts each. Okay, and I guess to give you a sense of scale, I'd guess the Artisan spans something like, probably 30 millimeters in length. And the Artisan contacts are about 6 millimeters squared in surface area. And the cylindrical contacts are 12.7 square millimeters. So the cylindrical contacts are 3 millimeters in length. So hopefully that gives you a sense for what these leads are like. The leads, by the way, are about 1.3 millimeters in diameter. So after the leads are placed in the patient's epidural space, they're connected to a trial stimulator. So one of the neat things about the SCS therapy is the patient can actually try the therapy at home before they sign up to get an implant, a permanent implant. So on the right here, we're showing the trial stimulator that is used. That uses essentially, with some minor modifications, the same electronics that the permanent implant uses. If the patient does well during the trial, then they'll be implanted with the implantable pulse generator, or IPG. This IPG, to give you a sense of scale, it's about a little over 20 cc's in volume, so about the size of a stopwatch. A clinician programmer is used to program the device and to assign, anodes and cathodes, and we'll talk a little bit more about this, or to put the electric field where you want to put it using the 16 contacts. Precision, I should mention, has 2 ports, each of which supports 8 contacts. And then there's a patient remote control. So the patient goes home and they're able to control the device. They use the same remote control for the trial as they do for a permanent implant. So during the trial, they get exposed to using a remote control and make sure that fits with what they're able to do. And then finally, because Precision was the first rechargeable spinal cord stimulator on the market. And so there's a charging system. It charges transcutaneously, and the charger is held within 2 centimeters of the spinal cord stimulator and charged. The charger itself has a rechargeable battery, so the patient can be mobile, so they're not tethered when they need to charge. So I'm going to move now to some of the highlight features of the system, just to walk you through them, and hopefully you'll get a taste of how we think about things in our group and at our division. So the hallmark feature of the Precision system we call MICC. Sometimes we use the word current steering, and the marketing folks come up with lots of creative names for this stuff. I'll go with MICC for the purpose of this talk. MICC stands for Multiple Independent Current Control. So on the right-hand side, you can see a high-level illustration. Each of the 16 contacts that's supported by Precision uses an independent bidirectional current source, is the essence of that. Where conventional stimulators use a single source, it may be voltage or current, but there's only one. So why an independent current source for each contact? So let's walk through maybe using a thought experiment to see why we believe that's important. If you use a single source and you want to stimulate near contact, say contact 2 in this example, you might assign contact 2 to be a cathode, and 100% of your current will flow through contact 2. And if it's a current source, you'll know how much current that is. And if it's a voltage source, it will depend on the impedances that are present. If you want to stimulate near contact 3, you would reassign the cathode to contact 3. And for simplicity, I'm not showing the anode, but we can imagine case or something like that. And if you wanted to stimulate both contacts 2 and 3, you would assign each of them to be cathodes. And the way that that's done with a single source system is the 2 electrodes are ganged together. And if it's a current source, you can program the total current, but you can't determine how much current's going through each contact, right? Because that's going to depend on the impedances, the relative impedances. So current will preferentially flow to the contact with least resistance. So a multiple source system like Precision handles this situation in the following way. If you want to stimulate near contact 3, you use one current source. And if you want to stimulate near contact 4, you use a completely independent current source. And if you run into the situation where you want to use both, you use independent sources. So there's not a branch node, and you can deliver precisely the amount of current that you desire to each of the contacts. And this has a couple of advantages. One advantage is it doesn't really matter what the impedances are or how they change subsequent to your implant. You're going to deliver the current that you prescribed with the programming system. A second advantage is that you can prescribe the current that's going to go through each contact, so you have much more control over the electric field. And in this little example, I've assigned 83% of the total current to one contact and 17% to an adjacent contact. And that allows you to fine-tune stimulation in a region of interest. So if we were to shuttle, the amount of current in different ways, we can move stimulation around, and we essentially have a virtual contact. Remember that we're living about 3 millimeters above the neural tissue in a typical case. Okay, so the multiple independent current sources are designed for better control of the electric field, which is important for being able to select the target of interest. And secondly, to increase your immunity to changes in impedance, which one would hypothesize would promote sustainability of stimulation. So I want to give you a little bit of an example of some clinical data so you can see how this might work. One of my former colleagues ran a study that we very creatively called the left-right steering study. And what you see on the left is 2 leads on a fluoro, and his assignment of the anodes is 25% each, okay? And then the cathodes were adjacent, and they were set to 0 and 100%. And then in random order, he presented to the patient different splits of cathodic current on these 2 contacts. So I'm going to walk through them. For the purposes of the demonstration, we unrandomized them and put them in the order such that the current flows from the right-hand side to the left-hand side. So if you see at 100-0 split, you'll get paresthesia on predominantly the right-hand side. This, by the way, on the right is a patient drawing. So the patient made this drawing. They had a little picture of a dermatome man, and they colored with the highlighter where they felt paresthesia. Now, if we were to shuttle 20% of the current, you can see the paresthesia start to move to the left, and 60-40 of the distributions that were presented to this patient was the one that seemed to fit most midline. Okay, so here at 60-40 split, we have a midline stimulation example. At 50-50, the patient's now experiencing most of the stimulation on the left-hand side. And you can see here on the front-facing dermatome man, a little bit of paresthesia that appears to have come from root stimulation. It was kind of neat, right, to see the anatomy and the physiology play out like that. You excite the root, and you get a band of stimulation at the level of the electric field. We continue to shuttle the current. It moves further to the left, and so forth. So this is exciting because we were able to generate a whole host of paresthesia distributions by changing the cathodic current on just 2 contacts. And if you have 16 contacts that can move current in a bidirectional way, there are myriads of fields that can be used and this allows you extreme flexibility in placing the paresthesia exactly where it needs to be, and importantly, trying to remove it from where it should not be. A third advantage of using the independent current sources, I'm going to illustrate with a video. And this little video is built from some animation, or let's call them frames from a finite element model output, and it's using an activating function-based estimate of stimulation. But what I mean by real-time adjustment is that under the normal programming paradigm, which you'll see on the right when I start the video, an electric field is generated by assigning anodes and cathodes, right? So they do some plus-minus programming, they turn the field up, and if the paresthesia, they ask the patient where the paresthesia is. If it overlaps with the pain topography, that's great. If it doesn't, then before they can make any adjustments, they'll turn stimulation off. Then they'll reassign the pluses and the minuses, and they'll turn it on again. They'll ask the patient. If it doesn't match, they turn it off. So you have this on, off, on, off, on, off, and you can get exposure to, I don't know, maybe 10 or 12 different stimulation configurations in a typical programming session. Well, when Precision came out, it started to push groups to look at real-time programming. In a real-time programming paradigm, stimulation is turned on, electric field is generated, and then if the paresthesia doesn't overlap the pain topography, or if there's a desire to move it, it can be done by incrementally modifying the position of the electric field in real time. So there's no need to turn it off. So let me just illustrate that with this video. And what you're looking at is you're looking at an AP view, or down on the dorsal columns of the spinal cord. And the little red blob there is an example of a pain target that I just threw in for illustration purposes, okay? So in this example, but the green is an activating function-based analysis. So initially with both paradigms, you would turn stimulation up. And again, our goal is to overlap the red with the green. On the right-hand side, if there's not overlap, you turn it off. And you choose a different plus-minus. Maybe you don't have bilateral stimulation, so you try minus on both sides. Or on the left-hand side, with the Precision approach, you're able to incrementally modify the stimulation, taking advantage of the independent current sources. And so this allows you, you can see quite a bit of flexibility, like we talked about before. And the patient is essentially being programmed by someone with a joystick. Sometimes the patient's given the joystick, and they just move paresthesia on their body until they get it in the right location. All right. I'll let this play through here for a second because it's almost done. So earlier, I suggested that there might be issues with impedance management. So I wanted to just walk through some examples of data that we've collected on types of impedances you see. And the first examples will be from spinal cord stimulation, and then I'll jump a little bit to DBS because that's where I've been spending some time recently. But on the left-hand side, you see an 8-contact lead. And the impedances were measured, and they're shown on the z-axis. Z-axis is in ohms. The impedances were measured on each of these 8 contacts with respect to the case at 4 different time points. And the time points are implant, 2 weeks, 3 months, and 6 months. So there's 1 trace for each. So you get this, I'll use the cursor here, you get what we call, if you will, an impedance profile. So this is what the impedance profile might look like at implant. And the impedances, you'll note, are not uniform, but rather there's areas of high impedance and areas of low impedance. And again, this is why the multiple sources are used to account for these.
Michael, when you talk about impedance,
Yeah.
where exactly in the current flow portion or potential are you making this measurement?
So the way the measurement is made is by using a short 20 microsecond pulse. And the pulse is of known current, and it's between 1 contact on the lead and the case. And at the leading edge of the pulse, we measure the voltage that's required to induce that current, and then we use an Ohm's law calculation to compute R. So that's how these impedance measurements are made. So it's not the complex impedance.
It's really an ohmic component that you're picking up.
That's correct.
At the leading edge.
That's correct. Yeah, I should have clarified that. The terminology that's used clinically is impedance, but you're right. It's an ohmic component. So this impedance or resistance profile, if you will, changes over time, right? So we can look at implant, and then 2 weeks later, that impedance profile has changed substantially. And notably, even contacts that are adjacent to one another can have strong differences in impedance, and where one was higher than the other at implant, they can switch which one's higher. So you can kind of imagine how the electric field might move in a case like that if you're using a single source system. Just to give you an illustration of patient variability, here's an example from a second patient that was collected by my colleague.
Is it current amplitude dependent?
It doesn't seem to be. It doesn't seem to be current amplitude, not for the ohmic component that we're looking at.
What range is the current?
So we use pretty small currents for this measurement. I'm going to be trying to make a recollection here. Let's see. I think we're something on the order of 0.8 milliamps, 800 microamps, something like that. So these are all intended to be subthreshold pulses at 20 microseconds at 0.8 milliamps. When you're this distance from the nerve, it doesn't excite anything, doesn't generate paresthesia. I'm going to switch a little bit over to DBS. This data that I'm showing was presented by Professor Alesh at the INS meeting recently in Germany. And what I'm showing is impedance on the y-axis for 8 contacts of a DBS lead, again at multiple time points on the x-axis. And this is summary data, right? So you can see, typical at implant might be something in the 800 ohms range with a standard deviation of a couple hundred ohms. And then it slowly increases. At least that's the trend if you look at the summary data. What I think is interesting is to look at examples from individuals, because that's what really matters, right, from the patient's perspective. If you're going to put 3 volts in the subthalamic nucleus, it's the impedance in that individual that's going to drive how much current is used for stimulation. So what I'm showing here are some examples of patients that moved in opposite direction that both contributed to the summary plot in the prior slide. But this gives you a sense of variability between subjects. So in subject 1, and again, this is a normalized to implant impedance change. In subject 1, we saw increases typically of impedance, not with some downward trends as well, but an overall increase. And in subject 2, we saw the opposite. And the changes were pretty substantial, actually. In subject 1, factor of 1.5, factor of 2 change in impedance. In subject 2, 30% decrease. So I think it's important to take a look at the impedances in individual subjects rather than just look at overall trends when you're considering possible impact. Now, all of that was done at a pretty long time scale. If we look back at this several weeks, right, out to a year. Many of you are probably familiar with this paper that Scott and Cameron wrote, where they looked at acute impedances in a DBS monkey model with a scaled down lead. And on the left-hand side, you see an illustration of the lead, and then 2 millimeters from the lead is a trajectory that a microelectrode was used, a track was generated, and at several points along the trajectory, the field potential was generated. So I call this the what the tissue sees experiment. And if we go to the next slide, they've plotted the what the tissue sees waveform at a couple instances in time using current and voltage control. So on the left-hand side, using current control, you can see that the microelectrode reading is very nearly the same at 2 time points, immediately upon stimulating and then an hour later. And in the voltage controlled, that's changed. And it's consistent with the plot that I showed in the prior slide. There's a decrease in impedance at an acute time scale. So in a matter of minutes, tens of minutes, the impedance has decreased, and consequently the stimulation changed. So we had seen things like this in some of our work, and it's one of the things that we're interested in now is, what are these acute changes, and how can they be manifest in DBS? So one of the members of my team, Carl Steinke, who's also a case graduate, proposed that we do the following experiment in a porcine model that we had been using. And Carl essentially wanted to mimic a monopolar review, which is the standard programming protocol for a DBS lead. And it goes something like this. You go to the first contact, you turn stimulation up, and you evaluate the clinical effects and the side effects at that contact. And then you turn contact 1 off, all right? And you do the same thing with contact 2, then contact 3, and so forth in a sequential way. And in his little experiment, he decided that they would use lower amplitudes at the back part of the lead, just to mimic that the best contacts might be the first few. Was the reason he did that. So at each of those time points or amplitude changes, he took an impedance measurement. Okay, so this is now pretty high resolution from a temporal perspective impedance evaluation. I'm showing normalized impedance, and you can see that as the stimulation was on, the impedance had an initial increase by about 30%. And then when contact 1 was turned off, so that contact 2 and the others could be evaluated during the monopolar review process, the impedance decreased. And it eventually decreased well below the baseline, right? So the net effect is something on the order of 40% change, 45% change. So here I'm showing the impedance profiles for the other contacts that were generated during the experiment. It also gives you a little bit of information about the spatial aspect of the phenomena, because you can see that when contact 1 was being evaluated in the monopolar review, the impedance on contact 2, which was inactive, was affected. And so that's another interesting phenomena that we noted. Now this is interesting because you might anticipate that it could affect things clinically. So if you're evaluating as a clinician contact 1 and then contact 2 and then contact 3, and let's just, for example's sake, say that contact 1 is our contact that yielded the best clinical effects, and you used, say, a voltage-controlled stimulator. So at 3 volts, we evaluated contact 1, but the impedance was high. And then at the end of the session, we decided to go back to contact 1, and we turn it up to 3 volts again, but the impedance is 45% lower than it was before. Then you're going to get a different stimulation volume with the final setting than you got initially. And in fact, when we've talked to some physicians, this concept has resonated with them, and they've shared experiences like, well, whenever I finish programming my patients with the voltage-controlled systems, I will, I'll send them downstairs to have lunch, and then I'll visit them one more time before I send them home, just to make sure everything's good. Now, part of that might be physiologic, but some component of it could be electric, as demonstrated in this evaluation. All right, so moving on, I wanted to spend just a couple minutes talking about a feature that we call Eagle Scan, or in our newer spinal cord stimulation system, it's called Lead Sync. And it's used to identify relative contact positions when you have multiple percutaneous leads. So the way that this feature works is something like this. So here's an illustration. So if we send a pulse to contact 4, again, a monopolar pulse, you can kind of imagine you might generate some isopotential lines. It's pretty rough isopotential lines, but let's just take it. Here's our isopotential lines. And then if we were to record at the same time the field potential on any of the other contacts, and let's just look at the contacts on the opposite lead, you would expect that the contact closest to the source on the source lead would have the highest field potential record, and that you would see some type of monotonic decay in each direction. And if you do this on the bench or in a patient, this is the type of thing that you'd see. Now, if one of the leads shifts relative to the other, and in spinal cord stimulation, this can happen because the leads are placed through this tuning needle, but they're anchored way back at the incision site. So, you have several inches of lead in the body, distal to the anchor point, and things can move around a little bit. All right, patients, when their pain goes away, they become more active, and before you have your encapsulation response, leads can move. So we can imagine that if the lead on the right-hand side has moved, and E10 is now the closest contact to the source, we would expect a shift in that profile. So you can use this, this type of induced field potential measurement to understand what the relative position of these leads is. And that's important to our programming paradigm. So I'm going to share some data from the same left-right steering study that we looked at earlier. So in one case, in each case, my colleague was trying to align the cathodes and come up with, in SCS, the clinical community calls this a guarded cathode configuration. But in this case, the fluoro was swapped. So he left and right were really right and left. And when he made the assignments, he accidentally put anodes and cathodes next to one another instead of cathode and cathode next to one another. He turned it up, and this was the paresthesia drawing that you see. And that shunting prevented this patient from getting a robust paresthesia response at a comfortable level. And further, the amplitudes were really high. As you can imagine, when you shunt like that, the amplitudes get really high. So my friend looked at that and he said, maybe I got this wrong. He went back and he corrected it, and this was the type of response that he saw. So we call this sometimes a parapants paresthesia response, which is a good, robust paresthesia response. And that can be achieved if you understand where the contacts are with respect to one another so that you can align the field properly. Okay, that's where the Eagle Scan, or the Lead Sync feature, if you will, comes into play. Recently, earlier this year, we launched a second generation spinal cord stimulation system, and that's shown here. So it's got all of the same components that we described before. On the bottom are all of the leads. Oh, good. Now you can see the 16 contact leads, which is actually really tricky to fit all of the cables that are required to drive 16 contacts in a 1.3 millimeter diameter sheath. You see the IPG. This precision spectra IPG has now 4 ports. Okay, so it supports 32 contacts. So you can have all sorts of combinations of leads. You can take 2 16 contact leads and place them in the epidural space. You can take a paddle and a couple leads on the side, all sorts of ways that the physicians can use these leads to accomplish their clinical goal. There's a new remote control a new programming system and a new trial stimulator. And so this is a platform that we put together. And I wanted to maybe walk through one illustration of some of the things that my team is doing. So there's an algorithm that's part of this new system that our marketing team calls the Alumina 3D algorithm. It essentially enables point-and-click programming and also compensates for this lead offset as part of the programming paradigm. So this is an example of how our neuromodulation research and advanced concept team, some of the things that we do. So one question when you have now 32 contacts in the epidural space is, well, how do I program a 32 contact system? You know, it would be difficult if you were constrained to assigning anodes and cathodes and doing it a traditional way. So this was the approach that we took. We wanted to develop an algorithm that took into account on the input sides the contact positions, and we thought we could get that type of information from either the user who sees the leads on fluoro or from the Eagle Scan or lead sync algorithm. Right? So we have the contact positions, and then we wanted to input some notion of the user's desired location of the field. So I just called that the desired stim field. And this is where the point and click comes into play. You know, they can choose a place, and that if we know what the field should look like and we know where the contacts are, we should be able to put together a field that will give them the desired stimulation. And the outputs are the percentage of current and the polarity of current on each contact. And because we're programming in real time, like we mentioned before, when you take a step, you want to make sure that you're not increasing the stimulation intensity or decreasing it too much. So you want to account for changes in the field that might change how the patient feels intensity. So that's also part of the algorithm. What I'm going to show you is a simplistic representation of the algorithm, but it's obviously a little bit more involved. So what we decided to do was use a model-based algorithm. So we had a finite element model of spinal cord stimulation, and we built what was called a reduced version of this model that would allow a priori simulation runs to allow us to take into account different contact positions and would estimate what the FEM would have given us out, right? So that's all in the programming computer. And we decided we could probably solve this with a least squares approach. So the contact position is all accounted for in a transfer matrix. The desired stimulation field is what I'm calling phi in this matrix equation. And J is the current distribution that we're looking for. And this is a well-posed problem as long as the contacts in the model aren't in exactly the same physical location. And as long as we represent phi with enough measurements that there's linear independence. So the user determines where they want the leads to be, or they push the Eagle Scan button and we tell them where the leads are with respect to one another. And that's part of the programming paradigm, and that's put into the model. And then the user defines where they want stimulation to be on that lead. So for our phi, what we decided to do is we would create a tripole or a bipole with the cathode at the position that they put the point and click, and we use the model actually to generate phi in this algorithm. Okay. And then the output, it's hopefully not too hard to see on the right-hand side, is the distribution of anodes and cathodes and their relative strengths. Okay, so in green you see cathodes, and in purple you see anodes. And what it's trying to do again is mimic a tripole that was run in a computational model using the contacts that it has available to it. Okay, so I'm going to illustrate this with a video. So you'll get this is a walk through the programming screens of our precision spectra system, and I'll just point out parts of the programming paradigm where this model comes into play. So what you're seeing on the screen here is what we call the lead configuration screen. So the user is able to select where in the anatomy they're placing leads along the spinal column. And they can look at a fluoro typically and do this. And then they drag and drop the lead into position. So here they're placing 2 of the 16 contact leads. And they'll drag and drop the port connectivity assignment. So it's at this point that we're starting to collect enough information to build our reduced model, right? We know where the leads are with respect to one another. You have to build a new A matrix every time that relationship changes. So that relationship's established on the lead configuration screen, and then you move to a programming screen. So here on the programming screen, the user is able to point and click, and that creates what we call a central point of stimulation. It's really the cathode location. And here they're illustrating, how the algorithm behaves when you point within the leads, within the contacts and outside of the contacts. And these distributions create then a bipole, in this case, that mimics a bipole in the least square sense in the model. And they can use up, down, left, right control like we described to move the stimulation up and down or left and right. And then there's a couple of other controls. I'm not sure all of them will be illustrated in this video. You can change, for example, we have a focus button that allows you to change the anode-cathode distance and fine-tune in a region of interest with even greater specificity than what we described earlier. So here the resolution's changed, and we're taking bigger jumps now. And what you do depends on what part of the use model you're operating in. In the operating room, you'll tend to move around pretty quick. You want to get to the general area. During a programming session, post-op, the patient will be given 45 minutes or an hour for fine-tuned programming. Okay. Whoops. So those are some examples of the technology that we work on at Boston Scientific and that's available in our product. I was asked to spend a few minutes talking about what does it actually take to go from a concept to a product in industry. And most of the time in life, the answer is it depends, right? So that's true in this case. It depends on the scale of the product that we're talking about, or the concept that you're trying to turn into a product. So let's, just for example's sake, consider a platform product like the precision system or the precision spectra system and all of the components that I described. So to give you just a very rough, high-level estimate of what that might take, you can imagine it might take something on the order of 4 to 7 years to come up with a platform, depending on how much prior work can be leveraged that is ready to go into commercial space. So it's been through any clinical trials or regulatory processes that it needs to get all of the appropriate approvals. And it's hard to take a guess on cost, but it would be in today's terms, several tens of millions. An example might be precision. So like I mentioned earlier, I moved to Cleveland in the fall of 1998. I think that's about the time that Dave Peterson and some of the other folks at Advanced Bionics were working on precision platform and architecting it and starting to find the battery that they wanted for their rechargeable system and all those types of things. And I graduated in 2004. That's about the time that precision was first implanted. This is the 2003-04 timeframe. So that gives you maybe a sense for the time scale to make one of these happen. And there's lots and lots of roles that are played in the process of taking a concept all the way to product. So what I thought I might do is just describe some of these roles briefly to give you a sense. And by the way, in a lot of these roles, at least in our division, and I suspect it might be this way in other divisions of other companies, the folks playing these roles have some type of a technical background, or there's biomedical engineers and electrical engineers and software engineers that are playing not just the R&D role, but our regulatory VP has a double E degree, this type of stuff, because it's important to understand if you're going to be able to articulate to regulatory agencies why something is safe and effective. It's very helpful to have that type of a background. So this also gives you a sense for some of the types of roles that biomedical engineers might play in an industrial setting. So just to maybe briefly talk about some of these roles, there's a lot that goes into understanding the customer, the workflow, exactly how a product is going to be used or is intended to be used, how it might be misused. All of these things are evaluated with some rigor, usually by someone from the marketing team. Lots of times we might have an R&D person there as well, just so they can get some firsthand experience. Marketing is also responsible for creating user manuals, a product launch plan, communications plan, all these types of things. And of course, this list of responsibilities and the list of roles are not exhaustive. R&D does a host of things. They define the product specifications based on the user inputs that come from market. They design the product. They do a lot of risk analysis. You've got to understand how each specification, if it were to fail, would impact the patient clinically. And then the rigor with which you test or with which you put in design backups and mitigations is based on the clinical harm that could be generated if that specification were to fail. Of course, it's all documented very rigorously. You develop test plans to ensure the product meets the specifications. Lots of times that involves building new tools to test the product. You have to validate these test procedures. And eventually, when you have a product in final form, you execute the test on the final product. I won't read these whole slides to you, but you kind of get a sense. There's a whole bunch of things that go into putting one of these products or platforms together. We have a quality assurance team that makes sure all of our processes are followed. We have a regulatory team that makes sure that we do the design and the development and the documentation per regulations. They're responsible for preparing submission packages and communicating with the regulatory bodies. The manufacturing group has a lot of work to do. They need to prepare a line that is able to handle maybe several thousands or tens of thousands of IPGs built in a robust way every year. So they have to have procedures and tools and receiving inspection and rework instructions when something gets messed up, and staff training at a high level of rigor, process validations, all sorts of things are required to make something like this work. We have project management folks that guide our projects. They handle all the schedules, the cost, the Gantt charts, the resourcing requests to senior management and so forth. We have clinical teams that are ready to a significant part of these investments is the clinical trials. They take a long time and a lot of resources, and companies like ours work really hard to do them right and have successes because they're large investments. And of course, once you have the product, you need to be able to sell it. And if I were to take a guess, I guess it'd probably be somewhere between, I don't know, 20% of our, maybe 30, somewhere between 20-30% of our neuromodulation division is folks in the field, sales folks that are moving the product into the customer's hands. So a platform like this is a big investment. There's a lot of roles to play and a lot of important things to do on all parts of that process. I wanted to, finish my talk just by telling you, one of the things that gets me excited about working in the neuromodulation space and in biomedical engineering in general is the patients. One of the great blessings, I guess, or opportunities for us as biomedical engineers and healthcare professionals is the opportunity to impact people's lives in a positive way while we work and have fun doing science and engineering. The person that I'm showing here on this slide, we were talking about last night at dinner, so I thought I'd maybe just share his story real quick. His name is Mike Roman, and he was an operating room technician. And he was playing basketball and injured a knee, went in for a standard scope to take a look at the injury, see what needed to be done, and he got a staph infection. And because of the staph infection, he eventually had to have his leg amputated. He ended up going through somewhere on the order of 30-plus surgeries because after the amputation, neuromas developed on the stump, and he was in a tremendous amount of pain. So the neuromas would be removed, along with another slice of his leg, and over a period of something like 12 years, his leg was removed all the way up into the hip, when there was nothing more to remove. And he was one of the recipients of our device, and right after it was implanted in the recovery area, he described the relief from the tremendous pain, and he was able to describe all sorts of interesting sensations that were just horrific that he had as part of his phantom limb experience and the neuromas and all of that, and the relief he was able to get was just tremendous and life-changing. And he'd been living on, lots of opioids to try and manage, which compromised his personal and family life in a tremendous way. And so once he got that pain relief, he asked us if there was anything that he could do. And it turns out it was around him that this campaign was built called the Race Against Pain, because he didn't want to go back to the OR, and he was trying to figure out what he was going to do in life. And he'd had fun as a teenager racing, so he went back into racing. But it's just really, really rewarding for us to have the opportunity to work with patients and hear their stories firsthand and watch them get emotional and understand that we had a role to play in that benefit. So I'll end on that note and take questions. Hi, Dustin.
Paraesthesia that you described, how did the patients describe that sensation? What do they actually feel?
Yeah, they have maybe some difficulty describing it since it's not a natural sensation, but something like a tingling or when your foot goes to sleep type of a feeling.
Is that pretty universal what all of them feel, or do you have variation?
I think that there's probably variability. Probably someone that works in the field more than I do would be able to give a better answer, Dustin. But it's usually along those lines. There are some parameters that you can change that will modify the sensation. Stimulation rate is the most notable one. So if you our default setting is, I think, 40 hertz. So if you stimulate at 40 hertz, they get something that's more or less comfortable. And if you turn it up, they often describe it being smoother. So they'll like that. Once you start playing with that knob, though, they want to go up. And what they may not appreciate immediately is that their charging burden for recharging the devices is going up pretty quick as well. So you bump it up to 80 hertz, they got to charge twice as often. So.
The last question related to this, does it tend to go away over time as they continue to use the device, or they always report sort of the paresthesia sensation?
No, they report the paresthesia. It doesn't seem to go away over time. No. And I don't know if that's because we've told them they should expect to feel it, and they turn it up until they do, or if it's that's a good question. I don't know the exact stability of the sensation over time if we were to leave it at a constant amplitude and a constant position, and to understand it at that level of detail. Yeah, Dr. Mortimer?
One of the things that catches my attention as you're speaking here, and I believe it's correct, but I would just need you to verify, that people coming out of the biomedical engineering department here at Case have had a significant involvement in there. You mentioned Dave Peterson. I don't know that people know he's a graduate here. It looked like that was Dongjo Lee in there. So, I mean, it's the number of people that have been involved, had the involvement here, and working with you guys that, you know, I kind of am proud of this crowd. I'd just like to see you, and I know, where's our chairman? He should be asking you this stuff. But
Yeah,
this
no,
is probably one of the, a significant factor that this department has contributed to that community.
Yeah, I can happily verify that that's the case. We've, in our division, we've had a number of Case folks, and I mean, I can mention, you're asking who, I can mention a few, hopefully at the risk of forgetting some, I hope I don't. But Dave Peterson was a student here, and he was the architect of the precision system and the multiple independent current control platform.
He was stolen from Medtronic by alumina.
Well, stolen's a strong word, Tom. But yeah, Dave chose to go and work with Al Mann's team. And,
He's gone again somewhere else.
He has, yeah. So Dave was with the company until a couple of years ago, and there are a number of others. You know, Rafael Carbonaro, who worked with Dominique, is my VP. I report to Rafael. I've been there for almost 9 years. Dongshuo Lee was there for a time. We have some of our master's students, Carl Steinke, Dave Blum, Mark Pierre's another PhD student that came out that worked with us actually a couple times. So there's and there's other Case folks actually outside the BME department that are part of our organization. So yeah, our global director of our DBS program, Deanna Harshbarger, I think was a Chem-E major here before she went to business school. So lots of folks from Case, and then we recently hired. I think you used the word stole yesterday too, Dusty. It's, Natalie Brill, who is probably someone that most of the folks in the room know. So yeah, every time we look for folks, we try and get variability, but there's definitely a core skill set here that keeps us coming back to look. So, and I'm glad that they did for me. Yeah. Yeah.
Could you describe your stimulator output a little bit? Is the anode phase capacitive, or is that constant current? And with all the flexibility that you have, it seems like you could easily exceed the Shannon curve if you wanted to. How do you manage charge balance?
Yeah. So The waveform has 2 phases, at least 2 phases. There's a primary pulse that's current controlled, and then at frequencies below 130 hertz or below, the second phase is passive. Okay, so and its duration is something on the order of 6 milliseconds. Once you start going to high rates and you don't have enough bandwidth, then we use an active recharge phase. And it's, if you look really carefully on a scope, you'd actually see a third phase where we do still do a little bit of shorting to pull the any residual current off of the output caps. But it's active biphasic, it's a symmetric biphasic waveform. The system's flexible enough that you don't have to be symmetric, but that's the way it's used today. With regard to the safety limits, we use standard safety limits for spinal cord stimulation. Usually those are defined in charge densities per phase. And for the DBS product, we also followed the lead of our competitors, which was to choose it's a charge density, but if you look, the charge density looks like it comes from a Shannon model K value of 1.75 or something like that. So we follow that limit.
Where were those let's call them standards, established? Are they grandfathered numbers, or do they come from Professor Mortimer? In his tomb. Where come? You look so good,
Yes, Scott even recognized it.
So I've been in the industry for 9 years, so I'll give you something that has maybe a little bit limited perspective. But I think that there's definitely a grandfathering component. These things have history, and where limits were established early on in the approval process, or in the approval history, if you will. And there's been safe stimulation, and those levels have been able to be used again in subsequent products. When there are novel products, then we get into interesting discussions with the regulatory groups, and try and sometimes we'll do preclinical experiments, for example, to demonstrate safety. We often use Shannon as a starting point for our preclinical evaluations on the DBS side. SCS has a lot more history, so and it does not have the context adjacent to the neural parenchyma, so it's different. So we treat the two franchises differently, but yeah, there's definitely some grandfathering. There's active discussion as for novel product.
So take sort of one example that's not at all simple, but it might seem simple, right? So you have in the same 1.3 millimeter diameter, now you have twice as many leads, right? So you have some sort of change of configuration of that material that's in there. It might be stiffer, it might not be stiffer. I don't know. It depends upon a lot of stuff you do. How much testing is required to get that lead, now your revised lead, through the FDA, recognizing that the sort of the external diameter is
Essentially the same.
essentially the same.
Yeah.
How does that work? Just as an example.
Yeah. So maybe I'll try and pick out some highlights, and if I miss any specific questions you have, feel free to ask them again. So if we use an example of the 8 contact lead to 16 contacts, that's actually a substantial design change.
Yeah.
And it took several years, starting with some prototyping, all the way through we start in a phase that we call technology development process. Before you even get to the product development process, you want to have your technical risk retired as much as possible. Right? So you go through this technology development process and try and work out all of the technical issues that you anticipate. And then the product development process ends up being something of a, it's when you go through all of the documentation, everything that's required by the agency, and all the formal testing. But yeah, as an example, the 16 contact lead, it's actually a little bit more flexible, has a little bit less metal, because the cables that were used to connect the contacts at the distal and proximal ends are a little bit smaller. So those cables are comprised of, in the 8 contact lead, 19 filar. I wish I had a cross section or a whiteboard I could draw, but essentially, if you were to take one of those leads in cross section, you would see a central lumen that is used for the physician to place a rigid stylet. Okay, and then around that central lumen, in the polyurethane substrate, are 8 individual lumens located radially around the central one, and then each of them houses a cable. So in the 8 contact lead, those cables are comprised of 19 filar. Right? And each of those filar has 2 dissimilar metals using a drawn filled tube process, one to promote mechanical strength and one for low conductivity or low resistivity, high conductivity. So I think we use a nickel alloy from the mechanical, and we use a silver core on the spinal cord side to promote the high conductance. In the 16 contact lead, we put 2 cables in each of those channels, each of the outer lumen, and the cables are smaller. They use 7 filar, I think, something like that. And then you go into the cadaver lab and you start testing these different concepts, and you see if it's stiffer or if it's not stiffer, and how the additional metal on the distal end plays into the use model. And you bring physicians in and you have them evaluate it, and you take fluoros and you track how an 8 contact lead behaves as it's inserted, and you compare it to how a 16 contact lead behaves as it's inserted, and you go through all of that kind of user validation part of the process. And then when you have all those types of things worked out and you think you have a product that you can start to put together, you enter this formal process development process, or what our company calls PDP, and you formally document all of the specifications. And once all of the specifications are documented and the team's working to design to them, like I mentioned before, they'll start developing tests for each of those specifications. As part of the risk analysis, they'll ask the question, what if this specification is not met? What happens to the patient? So it...
Is the FDA involved at any point in this, or are you guys developing all of these, like all of the specifications, assuming that you're going to meet some sort of a test from the FDA?
Yeah, we do it. We write all of the specifications. The FDA gives you guidelines on the elements of your process that need to be included in the submission package, and then we include those elements. For some types of products, our regulatory folks might have some early conversation with the FDA to ask them about certain risk elements and things like that, or to understand if a clinical trial is going to be important to them, those types of things. But for the most part, it's FDA asks you to have a process that's sound with certain elements, and it's on you to develop the process and use the process according to how you've described it. So I don't know if that gives you a sense. It takes there's a few years of tech development, and then 2 or 3 years of for a lead like that of development. Yeah.
How about testing? Once you get all that, how do you do the testing, animal testing, bench testing, reliability, performance? How many cycles do you test them to, or whatever? Do? Can you say some of those things?
Yeah, I'm not sure I'm going to have all of the right specifics, but we do, for certain types of leads, we do cycle testing, and we put them on, machines or robots to evaluate their robustness. It's especially important if you make a design change, like the example that we're using. You want to make sure that you haven't compromised robustness, or if there are differences, that it's not relevant to the use model, and those types of things. But the testing is pretty rigorous itself. You know, it requires design of tools and design of processes and tool validations and these types of things. I don't remember all of the specifics that are used for some of these tests. Where there might be a charge density question or something like that, or if we're entering a novel indication, we'll initiate an animal study as well, and maybe have some preclinical work to support our submission.
So all this effort and work begs the question of how many people you gave us how long it is and how much money's involved. Could you give us how many people are maybe full-time equivalent of the people that do the product development?
Yeah, boy, the folks that are the PM role would be able to give you a better answer than I could. But each of the roles that I described have at least one contributor on the project team. So when you get to the PDP part of the program, regulatory will have someone sitting at the table, quality will have someone sitting at the table. So these teams might be 8-10 people teams, but their managers, of course, are reviewing their work. There are people that are other people that are involved in the problem. So just manufacturing might have one representative at the table, but they have many people that are working behind the scenes to put the line together and do the training for the folks to manage traceability, receiving inspection of raw material, all that kind of stuff. So I don't know if I could give you a good number, but it's a large number of people that touches any one product design. Yeah, Kim.
All right, so as the host, I'm going to ask you the last question because we're almost out of time.
All right.
We're in this amazingly exciting industry, and it's growing, at a relatively rapid rate, and we're talking about billions of dollars, a year now, and so that's got to be a great thing from the perspective of someone involved in industry. But then, your competitors have recently had some pretty high-profile clinical trial failures in, indications that were, kind of like our expectations or Wall Street's expectations of the big growth opportunities. So, since you guys, could maybe have the benefit of learning from those mistakes, does Boston Scientific look at that as an opportunity, or is that something that becomes a scary proposition for R&D to have to convince your upper management that we got to keep pushing?
Yeah. I think that our organization doesn't see as positive clinical trial failures. There's lots of things that the groups work on. I don't want to say together, but you hope that they help that their activities are successful and help build a robust and healthy industry. And so we typically don't like to see those types of things, and the company definitely takes a look at them as we consider our own strategy and opportunities and things like that, and what the risks are. It might change how we view certain risks and things like that. But to give you an example, I think spinal cord stimulation, and again, I hope I'm not too far off, but I'll spitball a couple numbers just so you get a sense. I think it was something like maybe a $400 million worldwide industry when I joined the company. It's a little bit of a guess, but I'll just guess 400 million. And most of that went to one dominant company at the time, and there are now 3 active companies in spinal cord stimulation, or I should say large companies, and a number of startups. But with the 3 active companies, their combined revenue is probably on the order of, you know, I don't know, 1.2 billion or something like that. So, you know, a rough tripling of the market. So when you have, I think I said we had maybe a few hundred folks in the field. When everybody's putting a few hundred folks in the field, it can really help a given industry or a given indication, if that makes sense. It can really build it up. So in that way, the competition can be very healthy. I think it also, from you know, an engineer's perspective, it's great because it means everybody is continually trying to improve their product, right? So it makes it fun for the folks that are involved in R&D.